Thank you for a nice introduction. It is my honor to have an opportunity to introduce our Samyang Biopharm's DDS platform technologies in this the summit. And I am head of new therapeutics development business, PU. And Samyang Holdings was established in the year 1924. And revenue is over 3.5 billion as of year 2023, and total employee over 5,000. Our business areas is focusing on chemical, food, and biopharm and packagings. Especially Samyang Biopharm, we are focusing on medical device and also aesthetics, cytotoxic drug, and drug delivery platform technologies. The headquarters is located in the Jongno, Seoul City, and Samyang Discovery Center located in Pangyo, just a neighbor of this IPK. And we have a biopolymer manufacturing plant in Gongju, a southern area of the Seoul. And also we have two GMP manufacturing facilities. Both are located in Daejeon. One is for a medical device manufacturing plant. The other one is the pharmaceutical manufacturing plant. And we have a US the office in Cambridge. And also we have another GMP manufacturing site in Hungary. Okay, Samyang Biopharm's business overview and milestone. The medical device. We are the #1 global supplier of the suture, the threads. It occupies over 50%. So it is the observable surgical sutures, and also it covers the lifting threads, fillers, and skin boosters. And also second area, modified drug business. And we are the world's first company with biopolymer nanoparticle applying cytotoxic drug formulation. We have a cytotoxic anti-cancer drugs such as Genexor and Nanoxel. And also third sector, we are focusing on novel innovative drug delivery platform, which is called Sense. I'm going to go over more detail later. And it is tissues and organs selective drug delivery platform technologies. We have over 10 lead compounds. And also we are opening our innovation strategy for diverse collaborations. My talk will cover a general drug delivery system. It will give us some messages and also key points and pipelines in a sense. And also, I'm going to give you some the research result of the prophylactic vaccine development, which is a COVID-19 mRNA vaccine using one of the Sense platform, which is called ProSense. And also, next, the milestone of Samyang Biopharm, we would like to introduce our new application for gene therapy area, and also manufacturing capability and CMC support. And lastly, I would like to share some take-home messages with you. And gene delivery platform technologies. And gene therapy market is growing in precision medicine and also personalized therapeutics. So market size will be expected to grow steadily up to 52 billion by year 2033. And currently commercially available gene therapy drugs, for example, Skyvex and Comirnaty, which is the DDS was the LMP, and the Moderna and Pfizer, their total revenue was over $18 billion. And Zolgensma, which is the drug for SMA treatment, so DDS was the AAV9 viral vector. Casgevy, that is the SCD and TDT treatment drug. The DDS was the lentivirus. so which is the number first CRISPR-Cas9-based cell therapeutics. And this summary at the table designating drug delivery system. Why delivery matters more the therapies. So far, the COVID-19 mRNA vaccine prophylactic vaccine area, maybe LNP is enough. But for the further development area like gene therapy area, we don't need that. We need more advanced and the improved DDS system. So I'm going to go over with comparison. And please pay attention to the payload size, immunogenicity, and repeated dosing, and tissue specificity, and manufacturing and cost. And payload size, viral vector, liposome, LNP, maybe the viral, the SARS-CoV-2 spike full-length the antigen may be enough. But sense system, we formulated successfully from 20 base, like ASO siRNA, to 11,000 bases of self-amplifying RNA. And immunogenicity and viral vector definitely induced host humoral immune response. LNP, it is known anti-PG antibody response. Recently, there is some phosphorylcholine, which is the helper lipid induced antibody. However, the sense system, there was no any anti-PG and anti-DSPC antibody responses. Therefore, in terms of the gene therapy therapeutics area, it needs repeated dosing capability. So in terms of the cytotoxicity. So LNP, it has the anti-PG, anti-DSPC activity, and also it induced the liver toxicity. And viral vector induced the host immune response. So however, since there is no any anti-PG and anti-DSPC, therefore, our sense system can be applied for repeated dosing. And tissue specificity, viral vector, natural tropism, it infect liver, CNS, muscles. LNP, it is known, very famous to deliver to LNP. However, a salt system can be delivered into lung. However, the sense system, I'm going to show you, liver, spleen, lung, CNS, even spinal fluid can be specifically delivered. And manufacturing cost, viral vector, the large scale up and also the purification process is very complex, and also the cost is very expensive. LNP, we can generate with a large manufacturing module, and it is rapid, but the cost is high. A sense system cost effective. And this one is our schematic diagram of the platform. Sense is designating selectivity enabling nanoshell. So it has the lipid. In addition to the lipid, a polymer was added and hybrid system of the nanoparticle. So therapeutic payload, siRNA, ASO, mRNA, even self-amplifying RNA, CRISPR-Cas9 was successfully encapsulated, and ionizable, which allowing tissue selectivity, nucleic acid encapsulation, and also we added biodegradable polymers, allowing stability and safety. So we synthesized over 4,000 novel excipients designed and synthesized, formulated over 250 sense system formulation. And we obtained over 10 sense lead already. And there are four different key points in our sense system. Number one, no unwanted immune responses. So LNP sense-driven, the HE4 gene was delivered, injected intravenously with 5 microgram, 15 times weekly. And LNP showed a recognized foreign substance and triggering anti-PEG antibody response. However, since there was no evasion of unwanted immune responses. In terms of the HEP protein expression level, the LNP-driven reduced efficacy upon multiple dosing after 11 weeks. And however, since improved safety and long-term efficacy, I mean that the HEP protein expression level was remained. So, without ADA, sense enables a long-term treatment with superior safety profiles. And second key point, it has the lower toxicity, enabling repeat dosing. And with the same HEP mRNA was injected intravenously with two different DDS, sense and LNP, with 1 and 3 MPK. And weekly 5 times was injected, and then survival rate was evaluated. Even 1 MPK with LNP, there was a 20% the fatality was observed. However, sense-driven delivery showed 100% survival rate, even with a 3 MPK high dose. And in terms of the hepatotoxic index, LNP-driven the delivery showed abnormal blood biochemistry, safety level unfavorable for long-term treatment. Data was not shown here, but in sense delivery, the level maintained within normal range in terms of the hepatotoxic the index. So there was no toxicity or side effect induced by sense. So a superior safety profile appropriate for repeated dosing. And number 3 key point, application to diverse payloads. So we encapsulated from a linear left panel, linear mRNA and siRNA, shRNA, CRISPR-Cas, and right panel, the x-axis, that is the size of the payload, and y-axis, left side average diameter, right side PDI. From the 20 to 11 kV, as you can see, all the payloads was encapsulated successfully, and size and PDI was remained within our normal range. So stable nanoparticle formation with diverse payloads varying sizes. Number 4 key points. Our sense system is delivered organ tissue specifically, and this one is the passive transport. There is no any active active targeting moiety. So we developed F-look mRNA with 2 different routes, systemic delivery and nano-ready sense formulation delivered to spleen. Higher splenic dendritic cell was selectively delivered, and also it is efficient dendritic cell presentation, leading to potent T-cell immunity, which is applicable for anti-cancer therapeutic vaccine. And PanSense are located lung, especially endothelium cells, and it is efficient delivery sustained in disease setting. HepaSense, it delivers liver, highly hepatocyte selectivity. As you know, generally LNP deliver into the liver. However, depends on the disease onset. Some LNP delivery doesn't efficiently deliver into liver because that is dependent on LDLR. However, our HepaSense, the LDLR independently deliver into liver, which is the ASP siAllo-glycoprotein receptor. Our HepaSense is using siAllo-glycoprotein receptor. So that means even using our HepaSense for disease setting the target organ, it will be very efficiently deliver and also show the therapeutic efficacy in liver disease. And second delivery route, local delivery, ProSense intramuscular delivery, the muscle cell uptake, leading to a potent humoral immune response, which is very popular for a prophylactic vaccine. And PepSense, located to the lung, and especially pulmonary epithelial cells, and highly stable particle formulation inhaled through a nebulizer. According to the process of the nebulization, the mechanical shearing force is breaking down LMP. However, sense is very strong to be intact under the nebulizer mechanical stress. And neurosense delivered into CNS, and we delivered intrathecally, and it has the highest selectivity to brain, especially motor neuron, and also spinal cord. And this CNS, the study is ongoing. And this one is our pipeline, the summary. And the ProSense and NanoReady is ready for IND-enabling study. And HepaSense, PenSense, PepSense, we finished the non-clinical studies, and NeuroSense is in discovery stages. And let me switch the gear. I'm going to share some experimental data in COVID-19 prophylactic vaccine using ProSense. Unfortunately, our data, the manuscript is in preparation. I'm going to show just a summary. So C57 black 6 mouse was intramuscularly injected with 0.01 to 0.1 microgram, and twice, 3 weeks apart. 3 weeks later, a serum and also the spleen was harvested to evaluate the humoral and T-cell immune response. So result is that ProSense vaccine elicits a strong antigen-specific humoral as well as the cellular response comparable to the LMP. And challenge experiment, we challenged the human ACE2 TG mouse intramuscularly twice, 3 weeks apart, and 3 weeks later, we challenged with homologous virus lethal dose. And then 5 days later, we euthanized and we evaluated biomarker and clinical signs. And the result was the humoral immunity, including neutralizing antibody induced by ProSense vaccine, protects mouse from SARS-CoV-2 virus challenges. And immunogenicity of SARS-CoV-2 in NHP, we further evaluated reactogenesis, tolerability, and also the immunogenicity in a cynomolgus monkey. And 250 microgram of the antigen in 500 microliter was injected 3 times, 2 or 3 weeks apart. And then 3 weeks later, we euthanized for further pathologic observation. So the result was the ProSense vaccine exhibit excellent tolerability, reactogenesis, and safety even at high doses. ProSense exhibit no genotoxicity and favorable AD profiles. Please pay attention to this. Moderna, they used 200 microgram injection twice. From that study, even though we don't have the head-to-head comparison data, our sense-driven 3 times more higher dosing showed very safe result. Stability and liquid frozen storage stability at -20. It was stable as of over 12 months. And freeze-dried storage stability at 4 degrees, it was stable as of over 12 months, as determined by size, PDA, and encapsulation efficiency. And right now, we are going to evaluate at 18 months we're going to terminate after 24 months. In summary, process vaccine induces well-balanced T and B cell response, and also strong neutralizing antibody responses. And process vaccine exhibit excellent tolerability, even at high dose in NHP. And also lyophilized process vaccine allows stable storage at 2 to 8 degrees. And this one is our next journey and also milestone for further development. Basis of the Sense platform technologies, we are not stick to the prophylactic vaccine. We would like to develop the gene therapy, the therapeutics. So in one sector, protein replacement using antibody encoding RNA, which is the evading AER, and also cell and gene therapy, we are approaching in vivo CAR-X and also CRISPR-Cas9, and personalized medicine and new antigen anti-cancer vaccine can be applied. Those three sectors, based on the active targeting conjugation platform technologies, we are applying the cell-specific modalities using antibody sense. And this one is the manufacturing capability and CMC support. And we are supplying high quality sense key raw materials and delivering comprehensive IND enabling packages to support partners' property projects. So proprietary excipient GMP supply. As I mentioned, Gwangju manufacturing site, they are manufacturing and supplying the key raw materials for novel lipids and polymer. And non-clinical material supply, the process development for up to 300 milligram mRNA scale per batch for sense formulation established. And further is early stage of clinical trial material supply. We can supply clinical trial material in liquid or freezing dried or lyophilized format. And storage stability for over 2 years secured. So Daejeon plant, so it has the GMP facility as certified by the Korea, Brazil, Europe, and Saudi Arabia. And commercial manufacture for excipients and raw material for pharmaceutical products and medical devices. And we are open to the innovation and strategic partnership by out-licensing and co-development and joint venture. And I would like to share some take-home messages with you. And first, safety and adverse effect concerns for developing the therapeutics. And we need innovation in delivery system technologies. I think the DDS is a very critical and very important at this moment. And second, difficulties in production and supply. So we can improve in manufacturing and supply chain, depends on the process. And third, significant therapeutic expenses supply. So there is a resolution of cost and accessibility challenges. So we need to communicate, and also we need to solve that problem. Number 4, regulatory and ethical controversies. There is not many in vivo CAR-T using mRNA is not in the commercialized, and even neoantigen anti-cancer mRNA therapeutics, there is not any commercially available therapeutics. So early engagement with the regulatory authorities should be established, and also we need to initiate as early as possible. I would like to extend this opportunity to acknowledge our Samyang R&D team members and also the audiences. Thank you very much.
Thank you very much for your wonderful talk. So it's time for questions. Does anyone has question? Okay, yeah, Professor Barr.
Thank you. You seem to have achieved amazing results with your lipid nanoparticle delivery systems. Are you able to tell us how you've done that a little bit, how that you get targeting to different sites in the body, and how you reduce the reactivity? Is it just playing around with the lipids, or have you done something else, added antibodies, or can you tell us a little bit more?
Okay, good question. To introduce our history of the Samyang Holdings. Samyang Holdings is an expert, and we have a long history to managing, handling with a polymer. So that's the reason we are able to easily access to the polymer. And we have over 10,000 libraries of the materials. So to develop the novel DDS, which is called the SENSE, we used different composition and also different rates. So we, as I mentioned, over 5,000 excipients was synthesized. Among them, 250 was formulated and screened. So finally, we found passively transported certain specific novel SENSE formulation to 6 different target tissue and organs. So that is based on the efforts and also some experiments.
Yeah. Okay, thank you. Dr. Kamen.
For the interesting talk, actually. This is a very technical question, actually. In the lipid that you provide, actually, what's the formulation? Are you using ethanol? Are these lipids in ethanol? The reason of this question, you know it actually. If you use ethanol, there is that requirement for the scale to use explosion-proof environment. Can you comment on this part?
Yeah, as you know, the general LNP formulation, the lipids, full component of lipid should be reserved in ethanol. So our composition, ionizable lipid, same as LNP, and also we added some polymers, and depends on the composition and also rate. The buffer, reserving buffer is different. And also the key merit is that our formulation can be concentrated. So it's not really, in terms of as a matter of the safety of the manufacturing facility for anti-exploding the regulation. So there is no any problem. Yeah.
Okay, thank you. Any other question? Let me ask one quick question.
Yep.
So you explained about the tissue specific delivery using like different platform of your SENSE. And it is without any active ligands or antibodies. So can you explain like how it is possible?
I mean, it is very critical and also very openly questioned the questions. We don't know yet exact mechanism, but as you know, the serum protein, there are lots of different kind of serum protein, depends on the surface charge. So some specific the SENSE formulation component has the affinity to the certain specific serum protein, so it delivers preferably to the certain tissue or organ. So that's the known things.
Okay, thank you very much for your wonderful
Thank you very
talk.
much.